So I wanted to make sure that we didn't overlook the bulbourethral glands, and you can see that here just off of the spongy urethra. The spongy corpora cavernosa. So we can see that this is all connected to our urethra that's going to get us to the outside of the body. And we have the seminal vesicle that we talked about in the previous slide, the prostate gland that does all the above, and then the bulbourethral glands. This is going to produce this lubricant that contains mucin. It's kind of a viscous material, and this is going to be the material that is the pre-ejaculate material. So those are the three glands of the male reproductive tract, especially after we're leaving the vas deferens. All right? So now the penis, the portions of the penis, the root, this is going to be like the anchor. This is internal to the body. And so we can see that in the illustration, anchored in to the body. Then, of course, you've got the body of the penis. And once we look at the cross section of the penis, we're going to see that we're going to have three of these erectile columns of tissues. We have a pair and then an individual one. And then the tip of the penis is glands, and yes, there is no D. So glands, penis, that's going to be the head of the penis. And then there's the foreskin, and this is technically called the prepuce. In this particular slide, you don't see the prepuce here. There's no foreskin in this illustration. So as I mentioned, the cylindrical columns of tissue that are going to fill with blood during an erection, these are going to be the corpora cavernosa, and you really see these on, I'm not sure if you call this the superior surface or the ventral surface. We just say on top, especially as we look at the cross section. And then you've got a single corpora spongiosum that's going to be underneath those two. And within the midst of the corpora spongiosum, that's where we're going to see the urethra as it's coming to the urethral orifice at the tip. Now, if we cut this in cross section, this is what you see, a histological slide of cross section through the penis. These are the two large corpora cavernosa, and this is the corpora spongiosa, and here is the spongy or penile urethra passing through that erectile tissue. There's a lot of space, and you have a lot of valves in the veins of these that close off during arousal and allows the penis and those three erectile columns to engorge with blood. Those valves then relax after the climax, and that allows the blood to drain out. Yes?
So, the spongy urethra is not the dark?
No, not the dark pink. It's all of this area. This is the corpora cavernosa, and this is the corpora spongiosa, and that circle in the middle, and you can see this kind of dark purple, that's the epithelium of the lining of the urethra. Okay, and that's what that's going to be. But all three of these fill with blood and essentially inflates the penis. All right? Now, as far as the prepuce, the foreskin, it's going to be consistent with the skin that covers the whole penis. There's loose connective tissue around the columns, no hypodermis. The skin is thin skin. And you can see in this particular picture how it's like a hood that comes over the glands of the penis. And there's even a frenulum that is in this bottom part that connects to the glands and the underside of the shaft. And it's this, the prepuce or the foreskin, that is removed in circumcision. Now, there's been a lot of debate about circumcision because it's not the choice of the individual because you're a baby at this point. So, are there health benefits to having a circumcision? Some people say that it prevents infection. Apparently, you're going to have 10,000 cases of having the foreskin to find one infection due to having a foreskin. So all evidence really points to it being a social thing, a religious thing, a cosmetic personal preference thing. So if you have a boy, it's just a decision you're going to be faced with. And so understand, do your research, but there really doesn't appear to be as many health advantages to having a circumcision, I think, is what was once thought. Maybe we're just a cleaner society now. I don't know. Right. Any questions? All right. That completes the reproductive systems as far as male and female. Now what we want to look at for the remainder of our time is what happens at, and I guess a little before, but mostly at fertilization and once we get to implantation as we start developing the embryo. So I love this saying. Scott Gilbert is a famous developmental biologist, and this is a great quote, and this illustration, it's very complicated, and it's called fertilization. And to me, fertilization is an extremely complicated series of events. And the part that I particularly like about this quote says fertilization is the process whereby two cells, each on the verge of death. We're going to see that once ovulated, the oocyte has about 24 hours and it's going to die. Sperm don't live more than 72, most of the time 48. But they're on the verge of death and they unite to create a new organism that will have numerous cell types and organs. It's just the beginning of a series of cell interactions that characterize animal development. Now, a lot of people come to me as a developmental biologist and a biology professor, and they ask me the question, when does life begin? That's the whole abortion debate. I don't get into debate. I just use science because that's my job. I'm a scientist. And I tell them life began millions and millions of years ago. Now, that's not the answer they're wanting or looking for, right? But life began millions and millions of years ago. Life is a continuum. Because you can't take a dead egg or a dead sperm and get a living zygote. So life is a continuum. Whether by natural reasons or unnatural reasons, something causes life to stop. And that's why this quote is so powerful. Two cells on the verge of death unite to create a new organism. An organism that has never existed before in history. Your combination of DNA has never existed before. Unless you're an identical twin, and I guess then you are. Because you got a copy, a clone. All right, so we're getting to fertilization here. A lot of sperm, up to 6 million. Now there is some evidence that there is some chemotactic process. I think they found some genes in rabbits that can attract sperm. We know that single-cell organisms can find and follow chemical trails. Even our white blood cells can follow cytokines. But I don't think they've found a specific target for sperm to be directed yet. So essentially, one reason you have so many sperm is because half the sperm go into the wrong fallopian tube because their movements are random. So immediately, you're almost going to have the number of sperm available to fertilize the egg, because remember, you're only going to ovulate from one ovary every other month. And which one that is, the sperm aren't going to know. And since they're male gametes, they're not going to stop and ask directions. That was a bad joke. Okay. Somebody wants a better grade, right? So they're fast, 2 to 3 millimeters a minute, and some may reach the ampulla of the fallopian tube within 5 minutes, and some are lazy and take 45-ish. They stop off at Starbucks or Buc-ee's and they do their thing.
So they just they're just slower, there's nothing that's keeping,
Well, yes and no, and we're going to see what that is, and it could be. So where does fertilization usually occur? It has to be in the ampulla, and we're going to see why as we go through these slides today. The embryo has to be at a certain developmental stage to have created the precursor to the placenta, and that's what's going to cause it to implant. Now again, sperm may be 3 days, oocytes 24 hours. So again, when you think about the timing, again, it surprises me that there's ever any babies. Because you've got to get fertilization in the right place. That means the timing of the gametes getting together. The embryo has to be at the right developmental stage. The uterine lining has to be prepared to receive it. Yeah. That's why that first picture, the complicated picture, I think is a great illustration of fertilization. Now here's a term, and I've used it I think a number of times already in class, a zygote. This is the fertilized egg after its first cell division, where it's 2 cells, then it's a zygote. Once we start differentiating those cells as they divide again and again and again into more and more, then we're going to be talking about an embryo. But we haven't got there yet. Now, here's something I wanted to bring up, and it kind of gets to your question about, does some just take longer, does something slow them down? If you unfortunately have to experience the difficulty of infertility, and you go to a fertility clinic and possibly have to go through artificial insemination, so that means that your baby daddy, significant other, I don't know what you call them, your sperm donor in this case, they're going to have to provide a sample. That sample alone isn't necessarily going to be injected into your reproductive tract, because those sperm in that sample are incapable of fertilizing an egg, especially if you have to do in vitro fertilization, which is without a female reproductive tract. So sperm have to go through a process called capacitation. Now that's not capitation, which is cutting the head off, capacitation. And during this process in the female reproductive tract, and it's pretty, I don't know who drew this picture, but this says vaginal environment, and you've got some happy sperm, but there's one that's not so happy, because a couple of things have to happen. You have receptors on the surface of the sperm and other materials, and you have cholesterol on the surface of the sperm that have to be scrubbed off. And this largely is going to be because of the lining of the cervix and the vaginal canal. See, this is cervical mucus scrubbing to expose certain proteins. Calcium has to be present. That's going to be critical for the stage where the sperm's going to release its enzymes once it encounters the oocyte complex that's got the corona radiata and the zona pellucida. So all of these processes have to happen so the sperm then are capable of breaking through the barrier and fertilizing the egg. Now this capacitation process, it may take as long as 7 hours. So that does slow down the sperm. And some think that this capacitation, in fact, kind of prolongs the lifespan of the sperm. Because here you can see a sperm within some of these cilia that are present in the female reproductive tract. So capacitation is important in the process of the sperm reaching their final maturation. They've already been mobile. Once they're ejaculated, they're mobile. But now the head of the sperm is prepared to be able to fertilize. So we finally met up with the oocyte complex. We've got the oocyte here. we can see that the oocyte, it's got, and I wish they had not, again, they're trying to do too much in one illustration. But here we've got the second meiotic metaphase. Is that metaphase?
No.
What is that?
That's anaphase.
That's anaphase. So again, that's kind of driving you crazy. But we're at the second meiotic metaphase, and those sister chromatids, those 23 sister chromatids from mom in the egg, they're lined up at metaphase, but they're on pause. Then here comes the sperm with its 23 chromosomes, where we have to penetrate through the corona radiata and the zona pellucida. It's that white band that we've got there. And as the sperm comes in, the head of the sperm is filled with a package of enzymes called the acrosome. And when certain receptors on the head of the sperm encounter these cells on the corona radiata, that's going to release these enzymes that are going to help dissolve a path through the corona radiata and begin to allow the sperm to make its way through the zona pellucida. Now, the sperm doesn't completely digest all of the proteins and carbohydrates in the zona pellucida. Some of those are receptors that the sperm actually attaches to as well. And so that's going to be important for penetrating through. But as soon as the very first sperm penetrates the plasma membrane of the oocyte, the egg itself is going to change. And this is going to cause the release of enzymes within the egg that is going to convert those zona pellucida carbohydrates and proteins as being non-receptive to the sperm. And that is called the zona reaction. And the zona reaction prevents more than one sperm fertilizing the egg, which would be polyspermy. So again, one sperm penetrates, the egg goes, stop the rest. And it produces the materials needed to change the zona pellucida so no other sperm can attach, nor can they penetrate the membrane. Because it only takes one, it can't take two or more.
What if it does?
Embryonic death. It's more than one sperm. We'll think about it. What is Down syndrome? What causes Down syndrome? One extra chromosome of chromosome 21. Now, in fact, most extra chromosomes are lethal. Chromosome 21 is one that you can have severe birth defects, but is able to conceive and have a birth. Most trisomies, the embryo doesn't even develop very far at all. Imagine having an extra set of all 23 chromosomes.
Oh, okay.
And in Down syndrome, that chromosome 21, the extra one, what's wrong with it? Where's the mutation? It's a trick question. There is no, it's just an extra normal, healthy copy. But you can see the just debilitating effects phenotypically of a person with Down syndrome just by having too much of something that's good. So we absolutely can't have more than one sperm.
Maybe ignorant, but how are twins?
So we'll get to that as we get a little further in development. Twins, that was the question, right? We'll get to that in just a little bit. So fertilization. So the formation of the female pronucleus, this is that second meiotic division where we've got our sister chromatids all lined up, ready to go. It basically is like the end of mitosis now. I know that doesn't make sense. It doesn't make sense that, okay, we're in the second meiotic division, but now with fertilization, it's almost as if it switches at that metaphase 2. The rest is going to look like mitosis. And just hang in there with me, okay? Now, when the sperm nucleus enters, the sperm chromosomes are going to be replicated into sister chromatids, 23 of them. And those 23 sister chromatids are going to start lining up at the equator. What does that sound like when you line up at the equator? Sounds like metaphase. And so what happens is the pronuclei membrane begin to break down for the male pronucleus and the female pronucleus. Then the microtubules, the spindle fibers that are moving these around, they take the 23 sister chromatids from dad in that pronucleus that's broken down, the 23 sister chromatids that were in the female, the mom's pronucleus, and the microtubules are going to push them all to one equator. Now you've got 46 sister chromatids aligned at the equator, and that is exactly like metaphase of mitosis, not meiosis. Because now with those 46 sister chromatids in anaphase, we're going to pull 46 individual chromosomes to this daughter cell. These 46 are going to go to this daughter cell. Now we've got diploid daughter cells, 2 cells, and that's the beginning of the new organism. And now those cells can undergo mitosis and make more diploid cells that are going to become the embryo, the embryo and the placenta, and the various embryonic membranes that we have to have. I know it's complicated, so let's look at a picture. So in this illustration, this is what this is going to look like when the male pronucleus and the female pronucleus have come together and we're starting to pull, pull them apart, or is that too, where are we here? Oh wait, here we go. Let's go back down here. So here we have the female pronucleus, the sperm comes in, we replicate, and there's the male pronucleus. And you see how they get together here? They meld together into one. And now, although this looks exactly like this picture, they're trying to do too much at once here. Now we've got mitosis, we're starting to have cytokinesis, and we get 2 daughter cells that are diploid. Yeah.
Does the first fertilization provide another polar body?
No, and I don't know why they have these in here. Again, during the process of the division, this is just waste material, and it's actually outside of the oocyte. It's been spit out. So maybe it's within the zona pellucida, but I wish they hadn't left those in there. Do you have a question?
I answered it.
Okay, all right. Now these are actual eggs that have been fertilized. Here you can see a female pronucleus. Here we've got a female and a male pronucleus together. Here they're coming together, and here they've got the mitotic spindle where we're about to separate into that first division. And this is actually the tail of the sperm. I don't know how the tail of the sperm got into this one, but remember the tail of the sperm is filled with microtubules. That's the motor. And so that this stain with antibodies is staining for microtubules, and so they picked up the tail in that particular case. All right, so what does fertilization mean? The oocyte's going to complete that second meiotic division. Now the resulting cell is going to be diploid as it's doing its first division into 2 to be a zygote. Variation of species, crossing over, independent assortment, they've all happened and occurred. We've also determined the chromosomal sex of the embryo. Is it XX or is it XY? That's been determined. And that's going to initiate the cleavage division. Now the cleavage division, let's see, is it here? No, we'll get to it. The cleavage division is the first several generations of divisions. And it's different than mitosis and a regular cell cycle because there's no growth 1 or growth 2 phase in cleavage divisions. So the cell, the fertilized egg, is having itself, then those have again and have again and have again. So that's why you have to start out with a really large egg. because the first several generations, there's not a growth going to happen. So as the cells divide, they're getting smaller and smaller. Then at a later stage of development, there will be that growth phase and the cells begin to grow bigger. So the early divisions without growth are called cleavage divisions. Yeah.
Is that the most susceptible time for uncontrolled cell growth and uncontrolled cell division?
Is it? That's a difficult question, because if it is uncontrolled, that means it's uncoordinated, and you're not going to be developing the right structures, because many structures are going to be somewhat regulated by the neighboring cells and the secretions they do and that they produce, like growth factors, and it would just simply lead to a structure of cells that are not compatible with implantation or life, and I don't know how you account for that. Because it's just going to come out in the menses.
So because there's not like a growth stage of that, are they susceptible to becoming uncontrolled quickly?
I don't know, because you have your checkpoints and those breaks outside of the synthesis phase. They're like in the G2 and that pre-mitotic. So those would still be there. I don't know if they're controlled the same way even without the replication. Yeah.
The sex can be determined whenever the fertilization occurs.
Correct.
On the previous, like when we were going through female, we talked about the indifferent stages or the indifferent zones.
That's phenotypically what you see. Yeah. The chromosomes are still there. And again, the default pathway in early development is largely under the control of the X chromosome. The Y chromosome genes that have to stop the female pathway and turn on all of the chemical signals to create male, those are within the Y chromosome.
Okay.
And that's really all the Y chromosome genes do, and that's why it's so small.
Okay.
And there's so few.
Is that determined by the sperm or the egg or both?
What's that?
Like
It's by the
XY
male.
or XX?
Well, it's you know, yes, I'm reminded all the time that I have 3 daughters, and that's my fault, because you're either going to fertilize the egg with a Y chromosome bearing sperm or an X chromosome bearing sperm. Have we had the talk about choosing your gender yet? Yeah, and it's easy to do. You just centrifuge the sperm in a buoyant solution. So the X chromosome bearing sperm go to the bottom. The Y chromosome bearing sperm that are lighter, because there's not as much DNA, they stay floating to the top. If you want a girl, take sperm at the bottom, do artificial insemination. Or if you want a boy, take sperm at the top, you get a boy. I don't think they do that stuff for free and routinely, but it's possible. It's possible. All right. So here we are. Remember, fertilization happens in the ampulla. And you can see that here we've got our first cleavage division. We've got our 2-cell. We got our 4-cell. We got our 8-cell. And you see how this white, like the zona pellucida, it's not getting any bigger, but the cells are getting more numerous but smaller. That is characteristic of these early cleavage divisions. The zona pellucida is somewhat of a protectant of the early zygote as it's floating down the fallopian tube toward the uterus, and it's going to shed that and outgrow it by the time you get to the uterus. So here we are in our early cleavage division stages. And your first 2 cells, not the fertilized egg, but once it divides into 2, these cells are called blastomeres. Because very early on, when you're just a couple of cells together, you're called a blastula-phased embryo. Then after you have more cells, and I think it's about 18 plus cells or more, then you get to a stage that is called a morula stage. And morula comes from the Latin morus, which means mulberry. I guess somebody thought that this thing looked like a mulberry. Anybody ever seen a mulberry? I don't. Yes. I've never. Blackberry? Now, I've always wondered if it's like a blackberry or.
Would this be the more? That was AJ, and that was her brother.
Oh yeah, we're morula. Well, we may be a little further on, because we're not done growing yet. Now, you talked about twin. Who asked me about the twins? Okay. So at these early stages of development, especially in this 12 to 15 blastomeres, and even in the morula stage, the cells are called blastomeres. Maybe morulamere would be too hard to say, but they blastomeres. So all of these cells, 12 to 15 blastomeres, each cell is totipotent. Do we understand what totipotent means? If you could take those 12 cells and enzymatically digest them so they were individual 12 cells, and you could put them into a culture, like with in vitro fertilization, each of those 12 cells would continue to grow and become an identical individual. So identical twins are where early on some of the cells detached and broke away from the rest of the zygote. And while these cells are totipotent, they broke away and they just became an identical organism to the original. There's no way to tell which was the original and which was the copy, but they're the same. I'm sure with animals, not with humans, but yeah, I'm sure with animals that they can, because that's the only way that they would be able to say, oh, all of these are totipotent, if they had taken it in a lab, taken the cells out, put them back into individual, like maternal, females, and out comes a brand new mouse or rabbit or whatever, and when you run the genetics, they're identical.
What's the time frame of this?
Oh, this is we're still here, we're still in the fallopian tube.
But like, as the cells are splitting and everything like that, like, is it happening?
Oh, how long the divisions happen? Well, if we go back to this, I think this is day 8, day 6, 7, or 8 here when you get to the uterus. So this is going to be in the first 2-3 days after fertilization.
So they still need to implant?
Oh yeah, we haven't implanted yet.
So this could happen all the time, and then it just doesn't implant. And
Correct. Doesn't implant, you'll never know. And if fertilization occurs too late, then it's not going to implant, you'd never know. So there may have been fertilizations in your life that you just never knew. Because it didn't get fertilized in the right place, and so it didn't implant. Now, as we go a little bit further, these cells are going to begin to produce a hollow space inside the morula. And here you can see this blastocyst, and the blastocyst is really going to be the cavity here. And what happens is some of the cells will get segregated. We'll say this is toward the inside top of the developing zygote. Here you can see the green cells kind of circling all around, and then you've got these orange ones here pushed toward the top. Now we're differentiating what cells do. They're not totipotent anymore. The cells of the inner cell mass, that is going to become you. The cells in the surrounding shell, those are the cells of the trophoblast, and in Greek, this trophene, which we get the tropho from, means to feed. So the trophoblastic cells are going to become part of the placenta. It's the embryonic part. It's the part that's going to detach as the afterbirth, after you deliver the baby. So this is the trophoblast, this is the inner cell mass, and everything you are is going to come from the inner cell mass. So the egg lives how long? 24 hours. Sperm have up to how long?
72.
Well, you've got a 3-day window of implantation too. So this is why I'm like, how do babies ever happen? Because fertilization has to occur with the timing. In the ampulla, that zygote has to get to the uterus in time to be prepared to implant. So here we have the inner cell mass, we've got the trophoblast, and do you see these little kind of sticks out here on the side? These are going to be receptors. And there are going to be cells on the surface of the endometrium. This is the functionalis layer of the endometrium. And what they've discovered fairly recently is there's these little projections that are only around for maybe about 48 hours, and they call them pentapodes. These pentapodes are going to have receptors that match the surface of the trophoblast. And so you get this initial contact, and that's going to cause the zygote to attach. It's going to sit down, and it's going to anchor itself to the endometrium with this ligand receptor interaction. When that happens, the cells on the outside are going to begin to divide, and they're going to grow, and they basically form this large single unit blob that's multinucleated. You don't have individual cell membranes between all these cells. You see how this illustrates got 4 and 2? Well, it's really just one big blob that's forming around the trophoblast. On the inside, the trophoblast is still going to be single cells with nuclei. That's going to be the cytotrophoblast, cellular trophoblast. Out here, the ST, is the syncytiotrophoblast. So the syncytiotrophoblast, after you get the attachment from the cytocellular trophoblast, the syncytiotrophoblast is going to begin to eat its way into the uterine lining. It uses enzymes to digest and destroy the uterine lining. Now, the uterus is loaded with spiral arteries. And as this thing is digging its way and chewing its way into the uterus, you're breaking blood vessels. And so it's highly possible when you get pregnant, you may spot some, and that's going to be the spotting and the bleeding that happens from the implantation. But you should not have a full-blown period with that. Hugh.
Will you explain the interaction between the receptors and the cells of the functionalis again, please?
Yeah, so again, you've got ligands and receptors so that they link together. You see how you've got the green and the red linking together? That's how it's going to recognize that, okay, I've attached, I've landed. Now, this whole structure here is going to say, okay, now it's time to go on to make the syncytiotrophoblast.
Okay, thank you.
So does it dig into the functional layer, the basal layer?
Okay, here, this is just the cells on the very surface of the endometrium. In this illustration, if it was to scale, the basalis layer would be way down here.
Okay, so like an fertilized embryo really just implants itself in the functional layer.
Correct. Implantation is just in the functionalis, the functional layer of the endometrium.
Are those receptors only by the inner cell mass, or are they around the
Oh, they're around the whole thing, yeah. I know here it's just showing that they're around the whole thing, yeah. Yeah, because that ball could just touch and roll and, yeah. Anybody else? Okay. Now, notice what has happened in this picture. We have completely digested our way into the endometrium. And we've kind of covered ourselves over here with the endometrium. The only thing that's left after this thing has fully eaten its way down into the intramedium is just this little pimple-looking thing on the surface. That's the only indication that anything has happened. And yet here, you've got this big ball of cells that is growing and dividing and continuing to grow and grow and grow for 9 months. So this is what we're looking at in a different type of illustration. Here in the dark, dark purple, that is the syncytiotrophoblast. And you can see that these cells are going to continue to degrade the uterine, the functionalis layer. And you can see that we've broken blood vessels and we've broken through uterine glands. So the secretion of the uterine glands, the blood, is going to leak and spill throughout the syncytial trophoblast. Can you kind of tell how this is the earliest possible type of placenta? Because we're still just going to have diffusion providing the gas exchange and nutrient exchange right now. That's how small we are. But the syncytial trophoblast is going to give rise to the placenta. What's going to grow is going to be this inner cell mass in here, and you can already see that we've got this two-disc layer of cells. The top layer is going to be the larger, taller layer, and that bottom layer is kind of thin. This is going to be called the chorion. That's a freebie. That's the chorion. And you can see that we've got another space here that is going to be the amniotic cavity. Now, I think everybody's familiar with amniotic fluid, and the amnion is the membrane that contains that fluid. And towards the end of pregnancy, if you're starting to have contractions but you're not making progress, what is the OB probably going to break? Break your water. They rupture through the membrane and all the amniotic fluid comes out. And they say we're going to rupture the amniotic membrane. Well, it's really two membranes, the chorionic membrane on the outside, which was this, and the amniotic membrane on the inside. So they're really breaking the chorioamniotic membrane. But don't tell your OB that, oh no, that's not the amniotic membrane, that's the chorioamniotic membrane. Yes.
I think he has a first.
Oh, you're asking, okay.
I was telling him about
Well, I talk to him all the
I
time.
was just gonna ask, like, what if they did that early, like, could
Oh.
they have like side effects on
Well, just because you break your amniotic membrane and you release the amniotic fluid, that does not mean you're going to start contracting. Does not at all. And in fact, when you break that membrane, which is a protectant against infection, they're going to deliver the baby within 24 hours. Either you're going to do it or they're going to go get it. Your choice.
So we learned about that hormone, human chorionic growth,
Gonadotropin.
gonadotropin. What is the role in this?
So the placenta is what's going to be making that, really this syncytial trophoblast. Its role early on is to signal the corpus luteum, don't die yet, we're here. Don't die and kick us out, we're here. So that's what it's doing.
Will you point to the membrane again, please?
So the blue, that's going to be the amniotic cavity and the amniotic membrane. And this, I don't think they've got the chorion labeled here, but it's the yellow and that inner big kind of bubble.
Okay, thank you.
Yeah. Do you have one?
A little, I filled out that.
No.
So is it chorion?
The chorion is this cavity.
Is it a double membrane layer since it grows out?
Well, let's look at another couple of pictures. Okay. So now we're a little further in development. And what we've got here, our little grape, we've got the amnion growing here, this little bubble. This is going to be the yolk sac, which we don't keep around very long at all. And then here we have the chorion. The chorion's not going to grow an awful lot, but the amniotic membrane and the embryo is right in the middle of those two. That amniotic membrane is going to continue to grow, and it's going to push the chorion out with it. So the outer layer of the membrane is the chorion. The inner layer is going to be the amnion. Yeah, with the embryo situated right there in the middle. And here you can see we're further developing the syncytial trophoblast / placenta. And now you can see it's starting to push out on the endometrium surface. So that's going to get a whole lot bigger in 9 months. Now this, we've skipped a lot of stages at this point. But what I wanted to illustrate is the relationship between the syncytial trophoblast part of the placenta and the endometrium of the mother placenta. So the green that you see here This is going to be the endometrium. This is the functionalis. Here's the basalis layer in this lighter tan color. The pink, as well as these projections down, these are spaces. These are going to be your chorionic villi. And you can see you've got all these blood vessels coming down here in the villi. And in the cystisotrophoblasts, where we broke through and blood was just pooling in these spaces, well, maternal blood is going to come up into these really large spaces, and it's just going to squirt up into the space. It's under a lot of pressure. And then the venous blood is going to drain out. But what that blood is doing from mom in this space, it's going all around these feather-like capillaries in these villi, bathing them in blood and providing the gas exchange and nutrient exchange for the baby. And that's all going to go back to the baby or to and from the baby in the umbilical cord.
So in this scenario, how does oxygenated blood know to go to the arteries and deoxygenated blood know to go to the veins?
You talking about in here?
Like in the intervillous space, if it's just squirted up and then hopefully diffused by the
So
blood.
arterial blood's under a lot higher pressure.
Oh, so that's,
And so it's, you see it's shooting out like a fountain.
it's just a pressure gradient.
And it's pushing the blood into the lower pressure areas of the venous system. Yeah. So it just creates this circulation in here. The baby is pushing the arterial blood in the veins in that way. Yeah. Now, where in our circulatory system did the artery and vein definition about oxygenated blood, where was it backwards? The pulmonary circulation, right? The pulmonary veins were rich in oxygen. The pulmonary arteries were low in oxygen. We got another one. Look at the umbilical cord. Umbilical vein, oxygen rich. Because the vein transports blood to the heart. Well, we're picking up all the oxygen in the placenta, and we got to go back to the fetal heart. So here's another one where it's going to be flipped.
So you always take it in perspective of the baby.
Correct. In the circulation of the individual. In this case, the umbilical arteries and veins are part of the fetus, and so that's why it's going to be backwards as well. So, where does implantation occur? We know where fertilization needs to occur, right? Where? The ampulla of the fallopian tube. In my biology major classes, I usually have fill in the blank, especially my junior and senior pre-meds. On one of my fill in the blank exams, where does fertilization usually occur? My favorite answer of all of my 22 years, the eustachian tube.
What?
Eustachian tube. You know eustachian tube? Yeah.
That was a joke, right?
No, I am dead serious. I don't remember. I couldn't say anyway, FERPA. Yes, notice I said that. Eustachian tube. No, fertilization occurs in the fallopian tube in the ampulla. And implantation occurs usually in this upper portion of the uterus. Now, I think you can tell because of how large the babies get and the umbilical, not umbilical cord, the placenta is going to be pretty large. You don't want implantation to occur low. Once we get into the Oz of the cervix or down here really, really low into the cervix, it can cause some problems. So we're talking about implantation in the body primarily, maybe even higher up close to the fundus. But we don't want it low down near the cervix, because if it implants down close to the cervix, the placenta could grow across the opening of the cervix, and it could cause placentia preva. That's implantation across here, and you don't have as much to attach to, and it could lead to premature detachment. And depending on what stage the fetus has developed to, survival is all dependent on that. Because you really do want your baby at least 30, 32 weeks of development. 28 weeks development, we're kind of pushing it because the lungs haven't really adequately developed. And even if the lungs have, you haven't developed the type 2 pneumocytes. And what do the type 2 pneumocytes make that's important for keeping the alveoli open? Surfactant. Yeah, those cells are not going to come along until after 32 weeks. So if you have a preemie at 28, respiratory therapists are going to have to put exogenous surfactant into the airway of the baby. So if you get premature detachment because of that, that could potentially not be a very good thing. So I'm not exactly sure how far we are down the road here. We're probably 32, 34 weeks, I would say, judging from this picture. But you can see how much area the placenta covers. This would be just a normal placement kind of in the body, the upper part by the fundus, but here you've got the placenta coming over here over the os of the cervix, and that's placenta previa. That could be a problem.
How does the cervix not just dilate throughout the whole entire pregnancy?
Well, it's very muscular. I mean, it's very muscular, and so it is very thick. It's like a thick bottleneck. So when you start labor, you're not dilated, and the first thing that's going to happen even before the dilation, as the baby's head continues to push down, it's going to take the thick walls of the cervix and it's going to start to push them apart and thin them out, which is called effacement. So how effaced you are, how thin the cervical wall is, because it's really thick to resist the pressure of the baby growing and pushing down. Then once you fully efface, now it's going to start opening up.
What about your fallopian tubes? Are they just little?
Oh, they're just out of the way like everything else. Your intestines and your kidneys and everything just gets squished to the side because you've got this watermelon inside. So, yeah, hang on.
So why premature detachment of the placenta? Like, what does that mean exactly?
So typically, you deliver the baby, and then the placenta, the second birth, the placenta is delivered. Because the uterus doesn't stop contracting, it continues to contract and release the placenta, and that's the afterbirth. So if the placenta detaches before you deliver the baby, because how's the baby getting its oxygen from the placenta? Now you're in trouble. You got to have an emergency C-section and get the baby out quickly. Yeah.
How does the corpus luteum deliver or get estrogen and progesterone to the functionalis layer
Bloodstream.
through the blood?
Yeah, endocrine hormones.
We learned about this condition like EMT school, and like we were told like the biggest thing is to give mom oxygen
Yeah.
because like the biggest risk for the baby is that again that detaching early and then the baby not getting adequate.
Well, and you can understand too, that space there, the os of the cervix, you're not going to have any blood vessels coming up providing that to the placenta. So you might not have the same surface area for exchange of blood vessels and stuff from mom. So yeah, you just don't really want it there. Now, we also talked about ectopic pregnancies, fertilization that happens outside of a fallopian tube. And that can actually lead to implantation on the tube itself. Those are often called tubal pregnancies. And it can be a very, very dangerous situation because you remember the syncytial trophoblast? How does it implant? It digs in digestive enzymes. So wherever the zygote attaches, aberrantly attaches, the syncytial trophoblast is going to do its job. It's going to produce enzymes. It's going to start eating its way through. And if it eats its way through a major blood vessel,
That can cause like you not be able to have babies. Does that?
uh,
Oh,
yeah, that can cause you,
dead.
yeah. So if you have a tubal pregnancy and it does so much damage, you have to have your tube removed. I mean, that's one tube. Everything may be good on the other side, but yeah, if this attaches and starts eating its way through into a major organ and a major blood vessel, and you start bleeding internally, depending on that vessel, it might be like an aortic dissection, and by the time the EMTs get there or you get to the hospital even, your DOA.
How, you can go, I'll ask it after.
Okay.
How does that like happen? It's just like, doesn't have enough time to go down?
Well, if fertilization happens right out here in the infundibulum and the current or the flow just isn't right, or it gets in a turbulent area and it doesn't go into the fallopian tube, it lands somewhere else. Yeah. Yeah.
Okay, how, like, early would you know that you had a tubal pregnancy?
That's a good question, because you're not going to develop the placenta sufficiently to get the HCG to keep everything going, so you're probably going to have your normal periods. I think the first time you know it is when you're in a lot of pain. The pain is going to be the first clue, and it might be too late by that point.
Aren't there like certain IUDs or like what like increases your risk for ectopic pregnancy?
I don't know. IUDs lead to greater infection rates, I know, but I don't know about tubal pregnancies or extrauterine pregnancies. Yeah.
Are the fallopian tubes empty space, or are they hollow?
They have a lumen. I'm not going to say they're hollow, because you've got cells that have some ciliated cells, and then some cells have really, really short, I think, microvilli. So it's not just totally wide open empty space, but there is a lumen.
But there's no fluid or anything inside of them either?
No, there's not a lot of fluid other than secretions that the cells produce, and the current flow with the cilia moving and the peristaltic contraction of the tubes, because they are muscular, smooth muscle.
So that's what pushes the zygote along, is
Yeah.
like peristalsis?
And the cilia. Yeah. You made it.
Is this it?
That's all the information that I've got. That is the end of A&P 2 semester.